Welcome to Flow Stars, candid conversations between Dr. Peter O'Toole and the big hitters of flow cytometry, brought to you by Beckman Coulter at Bitesize Bio.
Today on Flow Stars, I'm joined by Garry Nolan of Stanford University School of Medicine, and we talk about moving outside your comfort zone.
I went there to learn biochemistry, something I didn't have. I mean, I always tell my postdocs and students, I don't think this is novel, go learn something you don't know.
Collecting carnivorous plants.
I collect carnivorous plants and strange plants. I like odd plants.
Reading for escapism.
I only read science fiction and fantasy. I'm only interested in stuff that's not real.
And UFOs.
Involved with this for the last 10 years since basically the CIA showed up in my office and asked me to help them with some of their pilots and ground personnel who'd gotten too close to what they claimed were UFOs and that they'd gotten sick.
All in this episode of Flow Stars. Hi, and welcome to Flow Stars. I'm Peter O'Toole from the University of York, and today I'm joined by Garry Nolan from Stanford, and also for many other things, which we'll get to as we go along. So Garry, hello.
Hello, nice to see you, and thanks for the invitation.
Yeah, likewise. It's been a long time since I last saw you, which I think was back in Glasgow, maybe in the UK, at Flow Cytometry UK, I think. I know I've been to a Cyto since then, but I can't remember. That I remember your talk really well from Glasgow, though, talking about Akoya and CODEX.
Oh, yeah.
Particular.
No, that's gone pretty well. They went public about 2 months ago on NASDAQ.
I didn't realize actually they hadn't already gone public, because I know a couple of the beta sites over in Belgium, Ghent, Leuven, combo, the VIB. So how's it going with CODEX?
Well, as far as I can tell, great. I mean, the whole point of starting a company was to be able to get all the quality control and, let's say, somebody to take care of the questions, the more routine questions on a daily basis. So all I know is that everybody who's got one, nobody's emailing me about the problems anymore, at least. And so that, to me at least, says, okay, they're getting what they need from the company itself. I know that their financials are doing great, actually. I'm on the board, so I can't say much more.
Yep. So I wasn't going to start here, but now we're here. Let's go with this, because in academia, there's a lot of people who are resistant to commercializing things and putting a cost to it. I think you've touched on some of the benefits already, but why is it so important that out of academia you still get spin-outs, commercialization of the products? Why is that so important?
So, if you really believe in your idea, right, and you think that it has commercial value, waiting around for the government to fund you is probably a big mistake, right? I mean, as a technology expands, as more money needs to go into it, at the very least, there's no money left in the NIH and our side, or I don't know what the various funding agencies are in Britain, but they just can't afford it anymore, right? And so, and at another level, a review committee would look at it and say, well, this is a risk, right? So we don't know that we have the money for that risk. Whereas if you go to commercial side, venture capitalists, they will look at it as a measured risk where there is a return for them, right? The NIH is not looking for a return. They're looking for a global return, public health benefit and things like that. But the VCs are looking for a financial return. And so they'll look at the individual and their track record. They'll look at the technology and it's what they think might be its potential and say, okay, well, there's a measured risk here. Here's what we'll put in. Right. And so there's way more money obviously available. I mean, I can drive down to Sand Hill Road, which is about maybe 5 miles from here, and it's on my way every day to Stanford. I basically drive down it, and there's probably $500 billion along that road, waiting to be somebody to talk them out of it. So, that's really the opportunity. And, I think it's, again, if you just put it on a library shelf and expect people to take advantage of it, that's a mistake. I mean, we all know, scientists all know that they think that their ideas are the best things since buttered bread, right? But actually getting it to somebody and convincing them and then getting it out there to everybody else, that to me at least is the satisfaction that, okay, other people are using it now. And that's the validation, I guess, that we're looking for.
Next, I guess with the likes of Codex, you could, I know, Heath Robinson, the setup in your own lab to a degree, although the probes are more difficult. But I think scientists in general, would you agree, are wanting off-the-shelf solutions so they can just do their science, answer the science questions, not play around, and they don't want to be engineers.
Right.
And
you come up with, buffer conditions and all the rest. So, that is what I was mentioning earlier about the quality control. You just want something you can hand out and it'll work again and again. And, people, I found, even when I was a student, I would much rather buy the NEB ligase buffer than make my own and worry that the ATP hadn't gone bad. Right. So, it's just, it can get too expensive if you overdo it. There's a little bit of, I think, self-ability that you need to work on, that not everything can come in a kit. But, I think that's probably the best way to look at it.
So I just thinking about the commercialization, would you say that you are actually saving the NIH money? Because a lot of your research and development funding and some of your lab members are paid by the venture capitalists.
Yes, we're saving money in the sense that, if you're bringing a new technology to the table, you're giving people an ability they didn't have before. So, if it would take 10 times as much effort to access the information that your technology can access with 1x, then obviously you're saving money, right? And then you're saving money on the far end with that you're actually providing more data per unit value. And that hopefully, at least in the work that I've always been interested in, translates into clinical applications. So if you're saving a single life, to me, that's invaluable.
So Codex, brilliant. Actually, I really do like Codex, the technology. I just love the approach, the strategy of it. But that's not the only company you spun out. Not your only. Yeah. So you've had CyTOF,
right?
MIBI, so multiplex ion beam imaging. Where did the ideas come from? Yeah, that's a lot of innovation and different innovations. Where do they come from?
Right. Well, I'll go down the list real quick. So Rigel, which is another public company, and it has a drug on the market, FDA approved. And I started that one when I was an assistant professor way back when. That was when, just back to your earlier point, that was when there was a lot of resistance. I was told I was ruining my career. There's a write-up of me in Forbes about how I'm some sort of evil, capitalist preying on, academia. What else was there? Then there was Nodality, which actually didn't do well. That was the only one that failed. Then there was Bina that we sold to Roche for 110 million. And then there was IonPath, maybe Akoya. What was the other one? Oh, Scale. It was another small company that I started that I also sold to Roche on the single cell combinatorial indexing that has been spun out again from Roche. 30 million was invested. And that's Jay Shindora, Cole Trapnell, and myself. And that basically is a single cell protein, RNA, and ATAC-seq simultaneously multiplexing. So it actually, the original intent of that was to replace CyTOF.
Yep.
Right? Because CyTOF has a limit, it's expensive. We can do CyTOF plus, we can do 100 parameters easily for 1/10 the cost And we already published that in Nature Communications last year, I think, or earlier this year.
So you're your own competition.
Yes, well, CyTOF, I mean, yeah, unfortunately, I mean, as useful as CyTOF is, it's expensive. And it takes specialized mass spectrometry knowledge. You know, I think it whet the appetite of the world, the immunology world, at least, for high parameter flow. You know, for many years, Becton Dickinson tried to beat it, promising me that they were going to crush it. Becton Dickinson didn't. So hi there. Still haven't beat it. And so, you know, I think to me at least, I mean, this idea came to me literally in a sort of a download in a talk I was at in Sweden. It just sort of fully formed. I said, wow, oh, that's a cool idea. You know, so we've taken it forward and it works.
How long to develop? So from that moment of, I've got an idea, this will work. How long to get from there to seeing the first results come off an instrument that's used for it to commercializing?
I mean, for the CyTOF, I mean, it was many years. I mean, originally it was invented by Scott Tanner, so I can't claim invention. Scott Tanner at the University of Toronto. He had already started his company, DVS. He came to me saying, okay, well, I'm a spectrometrist. You're an immunologist. Can you help me? And so, you know, I saw the value of it. And it was probably about 2 years for us to get the data for the first science paper that we had on it, the paper in Science that basically demonstrated the value and the utility. So it's usually about a year or two. I mean, CODEX from inception to our first instrument was about 6 months, but our first instrument was built with Legos, you know, to move stuff around, and it tape and glue, and you know, it was a mess. So it was basically 2 years to our first instrument, and then shortly thereafter the company was funded, and then it was another year to get it all ready to go in a beta.
That's remarkably fast. It is really, really fast. And to get it to market and to get in, or even if it's not fully commercialized, to get it to other end users, that is incredibly fast. If we look at the microscopy world, it doesn't seem to be that fast. Certainly the commercialization seems to take a lot longer. Is that because you've commercialized it with spin-outs rather than going to big companies to start with? Is it because you develop a company around it that it can be faster? Oh,
absolutely. Yeah, I mean, if you've ever worked within larger companies, I mean, I've not worked at a large company, but I've worked within many of them. There's this kind of emergent property of companies where the individuals are great people, but once it gets into a large enough organization, it just becomes molasses. And frankly, you have a lot of not too bright people end up running things, or at least they're inhibited in the kinds of decisions that they can make, or they're conservative. And so you can't get good ideas running in a big company unless it's something very unusual like Genentech and their research division, where that kind of creativity is promoted. And so I've always found that the smaller company, if you can get the right individuals, I mean, you can have the best idea, but the wrong individuals, and it'll all just implode. And so, you know, get the people. I usually try to move some of my students out if they're interested into it. And then I have enough connections in the area to know who's good and who's not to put in as you know, CEO or COO types. And then you just stand back. I mean, I'm actually not a micromanager at all. Ask any of my students. And so it's about learning how to trust people and not get in the way. I mean, one of the things I saw in one of my earlier companies, Rigel, was that when the company gets big enough, they see the founder as someone who's interfering with their decisions, coming in, and you know, you're second guessing them, and at say science meetings. And for me, I was never trying to second guess them. I was just offering my opinion, but they see it as a form of interference. So I found that you kind of have to let go and let them do their thing, and only interfere at the level of the board, not try to interfere directly, because it sort of questions their authority. And that's the best way to get pushed out of the company. So I learned that early on. And that's hard for scientists, especially if it's like your only idea, your first idea. You just don't want to see it go down the tubes. And, you know, commercialization research is a very different thing than academic research. And so you have to learn that difference. And a lot of scientists who start companies don't know the difference.
I can understand why that can be very difficult, because obviously you know it inside out. You've got the concept, you know where you want it to go, but maybe sometimes academics aren't the best business people either.
Yeah, well, yeah. No, they think that when they read how to start a company on the internet, now they're an expert. It's like reading how to do a protocol is not the same as doing it. There's a lot of art and zen to the doing of science as much as starting a company.
And as you say, getting the right people who've got the business side, which may be very different to the way an academic would develop it.
Yeah.
No, yeah, difficult times. How did you learn to sit back and not get involved with the sort of the fine details? You say it was just learning on the job? And
I mean, for me, it was learning, you know, but also, well, you have to be able to read a room, right? So if you're sitting in a room and you can see that the person who you've ostensibly put in charge of the project kind of fidgeting as you're talking, or you get a sense that they didn't like what you said, rather than attacking them, say, okay, what is it that I did that causes them to be, you know, like a hermit crab and pull back? And what operationally is happening politically in the immediate environment that you say, okay, well, maybe I shouldn't be here, right? Maybe I need to approach the problem from the outside rather than attacking it, you know, from within. And I think that I've always been a good read of people. And so a lot of scientists don't read the room. And so one of the big VCs that I worked with, Kleiner Perkins, once on one of my companies, the partner there that I worked with, when she and I would go out and talk to potential partners for the company, the first thing that we did is sort of as a debrief afterwards is, did you see how she reacted when you said that? She was a very good read of the room too.
Yeah.
And so I think there's a lot of that. That's how I learned, you know, was watching and listening to others who would say, Gary, step back.
So, and I presume you brought those skills into your academic life as well, because I can imagine the same mistakes would happen with anyone. So I think that advice is really insightful, actually, of how to deal with that.
You know, I was in David Baltimore's lab for my postdoc, right? And any immunologist, you know, who's been around for a decade or two would know of David. And so all the postdocs in his lab had been, let's say, their mentor's favorite student. That's how you know, and you were good, right? Presumably, you were good. But then you get a bunch of people like that in one lab all at once, and basically a bunch of prima donnas. And so it's a microcosm of the bigger world that you're going to walk into, where everybody is smart. And so you have to read everybody and what their potential agendas are, right? That you know, and not become a jerk, but find a way to navigate how to work with a lot of other smart people towards a common and positive goal.
So you mentioned working in David's lab. I guess I could be wrong here, but most famous for your innovations, your technology developments in the innovation space. And yet you started as a biologist. You know, David is a immunologist.
Biochemist, immunologist, yeah.
Yeah. So how did that transition occur? Is it a transition, or do you still see yourself as a cancer researcher, leukemia, immunologist. How do you see yourself?
I see myself a mile wide and an inch deep. So, you know, I was an immunologist in Len Herzenberg's lab. But again, I was his first or maybe second major, let's say, molecular biologist. But, you know, I learned all the computer stuff from Lee Herzenberg, and then the immunology from Len. And Len was an innovator. And I actually watched Len interact with a lot of companies, and I watched how he would do deals. He would bring me in to watch how he would do deals with other companies to get them to fund his lab. So I learned how to basically extract money from companies early on with him. And of course, Len had two of the biggest patents at Stanford, right? He had the flow cytometry patent, which brought in a lot of money. And then he had the humanized antibodies. So, I mean, that brought in literally billions to Stanford. And they made quite a bit of money too, themselves, the Herzenbergs. So in David's lab, I went there to learn biochemistry, something I didn't have. I mean, I always tell my postdocs and students, I don't think this is novel, go learn something you don't know, right? Go learn a talent. And so David's lab was biochemistry, and NF-kappa B and transcription factors were the big thing back then. And so that's where I went to his lab to do. But as it turns out along the way, we needed to deliver genes better to cells. And that's how I came up with the 293 T cell retroviral expression system, how to make retroviruses in 2 days rather than 2 months. And that just grew out of need, right? It was like, gee, I don't want to wait 2 months. How do I do this? Oh, I have this cell line that I know is really transfectable. Why don't we just try this? You know, and so that ended up turning into the Phoenix cell lines. And actually all retroviruses are now made in 293 T cells or 293 cells.
Yeah, super easy to work with as well.
Yeah, right. So, I mean, at the end of the day, I was still a better technologist than I was either a biochemist or an immunologist in David's lab, right? But it was, you know, I think it was a great step. I mean, it was also a great place for connections, right? I mean, David's lab pretty much peopled the immunology, you know, starscape over the years. And so, you know, you always had a bunch of people of similar mind around. And then again, watching people, watching the multiple different ways that you can be successful by looking at your peers and then looking at David, you know, and learning from him. I think I spent, you know, a total of 30 minutes one-on-one with David over my time. But I mean, he had 30 postdocs. You learned from him in group meeting, which was 2 or 3 times a week, because it needed that much time for 30 postdocs to go over their updates.
That's, yeah, but very little time overall then.
Yeah, but you didn't need it. I mean, you were surrounded by amazing people at the Whitehead at the time. You know, we were on the same floor with Richard Mulligan and the Weinberg lab, right? Bob Weinberg.
So, so you mentioned Len and his inventions as well, and how much money that made for Stanford Institute. How much comes back into the lab itself to fund new research? Well, for Len's lab, quite a bit. You know, so the way it worked at Stanford, if you have a, intellectual property or royalties, 15% comes off the top, goes to the Office of Technology Licensing to continue to fund that operation. And then the remaining goes 1/3 to the school, Stanford, 1/3 to the department, and then 1/3 to the inventors. And then the inventors will divvy it up according to whatever internal agreement they can manage to come up with. And so actually, in reality, none comes directly to your lab. It comes to the department, and then it's up to you to convince the department head that, you know, I should get this because of what I'm bringing. And so you might say, well, what's the threat? Why, you know, where's your leverage to get any money?
well, your leverage is, at least what I've always done, is if you don't give me this, I'm going to, the next time you invent something, invent it outside of Stanford, and you'll get nothing.
Yeah.
So
yeah, I certainly over here, it's quite hard to do that because kind of your contract is with the university, and you're kind of tied in with a lot of those inventions if you use a lab space to come up with it.
Yeah, I think it's, I think it's what Stanford did early on was realize that, leave it in the hands of the inventor, because they're the ones who really believe in it, first of all, and trust in their decisions, as long as you're not doing something which is essentially unethical or a real major conflict of interest. And, over time, like with me, I've built a track record of not screwing anybody over, right? I mean, my company, Akoya, two of my postdocs that we started it with, they're equal partners with me, right? I always make it equal partners. I don't take 90% because I'm the friggin' professor, right? Because that's just not, I don't think it's fair. Second of all, the postdocs are doing a lot of the work. And if I've got 5 companies going on, I'm partaking of all of them. So why do I need to be greedy?
It gives them an incentive, I'd have thought, to really deliver, because it's their, they've got a big potential.
Yeah. Well, with Akoya, I mean, these 2 guys are going to do quite well.
And hopefully carry on innovating themselves.
They've already started another company.
So there you go. So yeah, definitely innovating themselves, and the income comes back round. It's quite, you need that financial incentive, I think, back to the lab, back to the person.
Yeah.
To encourage them to commercialize it.
Yeah.
Back to where we started of, the why commercialize it. That income is vital for the university, for the departments, because it enables them it gives them breathing space.
Yeah. Well, it comes back indirectly as well through grants. So for instance, with Akoya or CyTOF and MIBI, I mean, there's many other investigators at Stanford who write grants around the use of those things. So, and I write grants around it. And so money flows that way. I mean, probably truly hundreds of millions of dollars have come in over the years because of the technologies that I've basically brought to Stanford. And I bring it to Stanford first. So there's a local incentive to say, okay, well, I'm going to get this established here first.
Yeah.
So we get to basically take advantage of it before our peer institutions do.
How much resistance do you get for the early adopters to start using the equipment?
Well, really not much, because so here's the reason why I've always worked with technologies, or I've found myself better able to work with technologies. Scientists usually conflict with each other over hypotheses. My hypothesis, your hypothesis, right? Who's right? I look at the stuff that I work on as what's insufficient with the kind of data that I'm getting right now that I wish I could do better, right? Because if I'm playing with the same technology with everybody, it's just, do my postdocs sleep less, right? Are we just, smarter or whatever? In general, I don't think any group is necessarily smarter than another. It's really what tools they bring to the table. So I look for what's inevitable, right? So what is inevitably the kind of data that we need to collect, right? So we might not have the ability to do that, but then you imagine what the perfect technology would be. I want to image and look at as many different markers as possible. That's a nice, mission statement, but there's no technology or practicality behind it. But then you start stepping back. Okay, how would I do this? How could I do 50 to 100 markers? Okay, and that's 50 to 100 antibodies. How am I going to measure 50 to 100 antibodies? You can't do it with fluorescence directly. So, and then you just go down that iteration path. And so what you end up providing then, having created that, and you go out and you publish a great paper like we did with Science or for CyTOF or MIBI or other things, other people look at it and they go, wow. And you always try to write the paper in a way that other people can see your paper as a window for their own success, through which they can view their own success. And they go, I can use that for what I'm doing and prove my hypothesis and beat that person, right? So basically you end up being an arms salesman. You're selling a new technology to both sides, and you don't partake in their argument. You just tell each of them that they can beat up on the other one better.
So you're funding both, you're arming both parties. Sit back and, enjoy the show. So huge amount of innovation, lots of work ethic. What do you do to unwind? What are your hobbies?
I play video games. Usually.
What's your favorite video game?
I used to play EverQuest. It's an old video game. It's an old one.
Okay.
It's just something that turns your brain off, but it's a bit of a strategy game.
Okay. What else?
I collect carnivorous plants and strange plants. I like odd plants.
So you've gone from arming research groups and now you're arming plants.
Arming plants. Well, they're just fascinating. They're beautiful. The carnivorous aspect doesn't interest me as much as just the unusual beauty of them. And, you look at them and you think about evolution and how did this trait evolve? And it turns out that the trait actually probably only really evolved twice. Almost all of the carnivores have a common root back in time. And as different as they might look today, they actually had a common ancestor that somehow, tripped upon the ability to stick insects to it and then devour them.
I wish I'd asked you actually to have one or two next to you so we could actually see a couple of these.
I could go out to the greenhouse and get one.
So how many have you got then?
I've got around 150 different species. Most, as it turns out, from Australia. Australia probably has the biggest collection of diversity of carnivores. And then a whole bunch from Borneo, and then others from scattered around North America, South America, and Africa. That's where they generally are.
And do you crossbreed these?
Yes, that's actually almost the most fun, to take two extreme forms of, say, the sundews, the Drosera, and cross them, and actually you sometimes get a viable hybrid. And that's the most fun to see. But the Victorians were crossing the Nepenthes. Those are the pitcher plants that kind of hang. If you go to, Kew Gardens, for instance, you'll see these Nepenthes in a large number of the big open greenhouses. I don't know what you call the large ones. And because they just were so unusual looking, the Victorians were really big into collecting them at the time.
So you're now comparing yourself to a Victorian, which is probably doing yourself a disservice.
They were very inquisitive, right? And recognizing things for their own beauty, right? I mean, they were busy pillaging the planet and bringing it all back to England, but sorry if I offend anybody with that. I'm British, so I can say those things.
I was about to ask, actually, what are your British roots?
I was born in Liverpool. My mother was a nurse. My father was an engineer on a ship. And so he got to go around and see a lot of the world. And that was when he realized that at that time, being from Liverpool was not going to be an upward mobility opportunity. And so he decided to move the family to the US. I think I was a year and a half when we moved.
Where did you move to?
Originally to Brooklyn. And then my dad had a job as an engineer, basically power services. And then we moved to Louisiana and Seattle. We moved around a lot, trying to find a place where my mother, as it turned out, was allergic to mosquitoes. So Louisiana and Seattle were out.
Yep.
So we ended up settling in Connecticut, which is pretty much where I grew up.
How old were you when you settled?
I think 5 or 6. Yeah, it was kindergarten. So 5 or 6 by the time we
A
settled.
lot of switching around, but settled at a school age, I guess.
Yeah.
God. So if you could live anywhere, where would you live in the world?
Kauai.
Okay. There's not many coniferous plants there, are there?
No, but I'd have to bring them all there.
What's the attraction?
I like tropical areas. It just smells good.
Okay.
It's just, there's a lot of other places, but you're really talking where you want to retire. That's where that's one of the places. Spain, near Barcelona, right? Portugal. I actually wouldn't mind moving to Ireland or having a place there.
Okay. Well, you've just gone from quite warm, tropical or warm areas to Ireland.
Yeah.
It's not the warmest.
What's the quote? If I contradict myself, I contain multitudes.
Okay. And you've got Irish roots as well.
Yeah. Mother's maiden name Murphy, Nolan. Those are like 2 of the most common Irish names.
Yeah, even more common than O'Toole, I think, for it.
So what was interesting is I went back and looked, following the roots. The Nolans have somebody, I don't know who did it, they have the largest genealogy and actual genetics tracing. They have there's a web page where all these Nolans have collected the genetics and following it on the X chromosome, the particular RFLP on the X chromosome, and they can trace it all the way back, and then they've traced it off to somewhere in Uzbekistan is where the original mutation came from.
I guess probably most of us probably originate around that area from a European perspective and European roots. Who in your career do you think has been, this may have multiple answers actually, who's been your inspirations inside and outside of work?
Well, I would say inside work. My 3 mentors, Len and Lee, for different reasons. David Baltimore for the height of focus that he had on things. An earlier mentor as well, this guy Aladar Zaleh, with whom I still work. He was my undergrad mentor, and so he and I work pretty closely still together. He's actually taking a sabbatical in my lab. He's from the University of Würzburg now. Outside work, it would be people like, you probably don't know him, Jacques Vallée, and he's mid-80s and he's a very good friend. And a guy by the name of David, no, not him, John Mack. And there is one of my outside hobbies that I follow.
Okay. And who would you most like to meet if you could?
Richard Dawkins.
Okay.
I just love his stuff. And who's the other one? The Roger Penrose, right? Just won the Nobel Prize for quantum physics, but is really, at least his work is beautiful from quantum physics, but he's been the person most pushing the notion of quantum consciousness. The notion that your consciousness is not, between your neurons. Your consciousness is actually computed in your microtubules because they have a quantum signature, and that's where quantum computation occurs. He got a heck of a lot of flack for the idea, but he's been the biggest proponent of it, and it just makes sense.
So, thinking, I did look at your CV and it said that you're an outspoken proponent, thinking of proponents, of translating public investment into basic research for public welfare. Mm-hmm. Why outspoken?
Well, because back in the 90s, it was something that you didn't do. back when I was starting, I mean, you were dirty for, for touching commercialization. And it, gets back to that term. It was like, to me, it was like inevitable. You watch these funding cycles at the NIH go up and down, and yet commercial opportunity and commercial monies were always available, right? I mean, I could sit down, because I had developed a 293 cell system. When I arrived at Stanford, everybody wanted to work with me, including a lot of commercial entities that were gene therapy. And I could convince a VP who could write a check for $250,000, which is basically an NIH grant that you traumatize yourself for 2 years to get. I could get it in with a lunch, right? So, duh, right? So, but there was still this constant pushback. So what I learned early on, because I went and got permissions to do this, and I got a track record written down. This is before Stanford even had a dean of conflict of interest. Sorry, I use air quotes all the time. It was, I got all the letters to make sure I was doing it right, so that when people came and accused me of things later,
yeah,
I said, look, I've got all the permissions. The deans have approved this with the following boundary conditions. So go away, leave me alone.
Do you think it's still such a dirty? It's not such a dirty thing anymore, is it?
No, but it just comes from knowing that you're right. It's one of the things I tell my students. I said, if you know you're right, absent it being illegal or unethical, do it. Don't listen to people who tell you you shouldn't do it, right? I said, because really what they're doing is they're using the most primitive form of control. They're using shame to make you do something that they want you to do or not do. And that if you really believe that what you're doing is right, do it. Don't let anybody stop you, because if you do, you belong to them.
So this is really back to reading people and understanding motivations and the people themselves. So it's fascinating. Yeah. So on that balance of using public investment for basic research, how do you balance the importance of publications over patents?
Oh, publications come first. It doesn't, you can patent something well before the publication is out. You can put the patent in, but I never let patents or commercial stuff get in the way of a paper. I continually have even conflicts with all of my companies over the years where they say, well, we don't want you to publish that. I said, no. We're publishing it. The other sort of adage that I always say is, if you stop somebody from doing something or try to stop them, they will invent a way around you. Don't give people a reason to invent a way around you. If you give them the information of how you made that buffer, they'll buy the buffer rather than inventing their own.
Yeah.
Because the moment you invent your own, somebody invents your own, then they're going to find a way to commercialize it around you. So just give it to them, and because people are inherently lazy. They want to get something which works.
Is it lazy, or is it just wanting to speed up the process? It's that arms race again, isn't it?
Yeah,
don't invent the weapon if you can buy the weapon, because you want it in the arms race. You want to get to the result quicker.
Yeah.
Some quick fire questions. Mac or PC?
PC.
Early bird or night owl?
Night owl. Both, actually. But I get more work done at night.
Yeah. Why do you think you're better at night than the morning?
It just seems quieter.
Okay.
Sort of like the mental ether just seems quieter. You're less likely to get interrupted with an email that you see urgently requiring you to do whatever.
Okay, so things are just your space more than being interrupted. Yeah, over the past 18 months or so, what do you find better, virtual or physical meetings?
Oh, definitely still physical. I mean, I got to go down, actually my first on a plane trip was just last week down to Santa Barbara. I'm part of the Sean Parker. He started Napster and was a co-founder of Facebook, and he has a Parker Foundation for Cancer Immunotherapy, and he's got a lot of the immunotherapy people involved, and I'm there for basically technology. And so he pays for us to go to some very nice places twice a year when we were allowed. And this was our first one down in Santa Barbara. It was wonderful to finally be able to do things again in person. Although I have to say, I'm a bit of an introvert. And so I like being alone. So the last 18 months weren't really that bad for me. I mean, Zoom is not my preferred mode of interacting, but not traveling was definitely a joy.
Did you find you had more time, or actually things just got busier?
No, plenty, lots more time. Lots more time to focus, and no, it was fantastic.
Okay. What's your favorite food?
Hamburgers. Curry, actually. I have to say, no, I'll take that back. Curry.
In America or in Britain?
Britain. No. When I go to London, I head straight for the local, whatever the closest decent curry restaurant.
Yeah.
Best in the world by far.
What is your what food do you least like? If you were to go taken out for one of these meals and as soon as it was put in front of you, you think, oh no, I really don't want that.
Any kind of fishy fish or lamb. I like white fish. I don't like fishy fish. I can't eat lamb. It's too gamey.
Cool. I'm with you on the fish.
Anything that I can see or taste and know what it was, if it was alive, that bothers me. So I can't handle bloody food. That reminds me that it was alive. Hamburgers, because they're all mashed up and you have no idea what it was.
So your steak is always well done then?
Yes, crisped.
I've got to ask, being you like hamburgers, what do you have on it? Just ketchup, mustard, barbecue sauce, cheese,
mayo, and pickles. I don't like the tomato and lettuce on it.
Just too much faff?
It's just too much. It gets in the way of the real thing you're trying to taste. Just
totally get that as well. So would you stop off at McDonald's for a quick eat?
Oh, yes.
McDonald's or Burger King?
McDonald's.
Yeah, good man. Right answer.
Yeah.
Definitely. And is that a Big Mac quarter pounder or just a hamburger?
Quarter pounder and fries.
I do like a hamburger, just a plain. In fact, no matter what I have, my chicken sandwich, a wrap, whatever else, I always have a hamburger just on the side.
Yeah. I mean, I have to watch it. I mean, I managed to lose 30 pounds over the whole COVID time. So it's, the not traveling has really helped. And I've gotten myself down to target weight.
So is that just a healthier diet, or have you been exercising?
It's both. It's actually not drinking milk, of all things,
okay.
And only eating breakfast and dinner, and just going without lunch.
You drink milk when you travel?
If it's available. I won't anymore because it was. I mean, the reason I got into milk was because when we moved to Connecticut, the water when I was a kid tasted awful. It had so many additives. It was just like, how could anybody like this? It was gross, and so. We just got milk, and that was my standard. And it wasn't until I moved to California that I realized that actually water can taste good.
Or not taste much.
Taste at all, but at least, yeah, can be refreshing without, the chemicals.
See, the thought of milk in other places that may not be ice cold or might not be really fresh.
Yeah.
Nah, I don't do milk outside the house.
I mean, that stuff was deadly with me, and my grandmother would send us from England these care parcels that were usually filled with Cadbury's chocolate, right? And I have to say, back in the 80s, I think the chocolate was better, I don't know what they've done to it. At least here in the United States, it's just, they've turned it into Hershey bars. It's gross, the stuff that they make. So, but chocolate and milk are deadly for me.
Okay. So next time you're in the UK, I will get you the Cadbury's chocolate.
Cadbury's chocolate.
Cadbury's chocolate.
Yeah.
Dairy milk. It's got to be dairy milk, hasn't it, at that point? Okay. Tea or coffee?
Tea to get up, and then coffee midday.
Okay. Wine or beer?
Wine. I can't drink beer. I don't know why. It's just something about it.
Not even a stout.
I like certain British beers, the warm drafts.
Yeah. Ales, bitters and ales.
Bitters, yeah, brown and bitter.
So stout, Guinness, Murphy's.
Guinness is a bit too strong for me. I mean, it's good, but I could never get drunk on beer. Martinis, gin martinis, absolutely.
Not espresso martinis, then.
Espresso martinis, what's that?
Yeah. Oh, really?
They put espresso.
Coffee liqueur, so Kahula, generally.
Okay. All right, I'll try it next time.
Yeah, no, okay. Well, now I owe you two things. So I'll actually get you the Cadbury's chocolate, and you can take it down with an espresso martini. Red or white wine? You said wine, so red or white?
Red. Red.
American?
I mean, there are some good California wines for sure, you know, but you can't beat the French on wines in general.
So you've had a lot of time at home throughout it. What would you rather do, read a book or watch TV?
Read a book.
Okay. What sort of genre would you?
I only read science fiction and fantasy.
Okay.
I'm only interested in stuff that's not real.
So in that case, what's your favorite movie?
Thor Ragnarok.
Okay. Good fiction.
That's I probably watched that about 80 times. Or Aliens, the second of the Alien franchise.
Yeah.
Sigourney Weaver.
That Star Trek or Star Wars?
Star Trek.
Yeah, absolutely.
Star Wars is, I mean, it's okay, but it's just too, I don't know, just a little too childlike.
So coming through to more serious stuff again, it's deviated off that, but it was fascinating.
I'm sure now like some intelligence agencies have like a total profile of me now.
Well, I don't know, but any conference organizer inviting you over, they know exactly what they've got to give you to drink, to eat, to read. If you could change one thing in scientific research, what would it be?
Well, the review process, which is inherently broken more in the grants arena than it is in paper review. But I mean, I'm not the first, obviously, to say that it's broken, but the grants especially. I mean, the NIH, it's just awful. I sit on grant committees all the time, and you know, I don't think a lot of grants get a fair shake. And all it takes is one person to give it a low score on a board of 50, and that's it. It's just sunk. And there's no amount of arguing that you can do that doesn't come off as combative. That's going to basically bring a grant back into the fundable range. And I just have always felt that large committees are wrong. You know, small committees are what you need. You're more likely to come to consensus with a group of 5 than a group of 50. And so, you know, I remember I was put in charge of one of the large program projects at the NIH to, you know, get it funded. And they said, okay, what do you need? I said, I need the following 3 or 4 people, and we'll pick the best. And then they said, okay, well, that's good. Here's a list of 50 people that will be on your committee. And I just said, this is a disaster. You know, it just turns into a disaster.
I take it at that point. So I sit on the UK funding panels. You say it's big. Does that mean there's not much discussion because there's 50 people? So actually people tend to talk to the ones that they've been charged to talk about, and very few others contribute at that point?
Yeah, well, they're not, how can you, right? I mean, they're not all given an opportunity to talk. And, we all know this. Some people are just more talkative than others. They all think that, and they become the dominant voice in many of the decisions. And that's just wrong. Because there's plenty of smart people who just don't like to talk because it's a large group. And so I don't think those large groups really pay much attention to the psychosocial environment and context of what happens to humans in larger groups. And so, you're basically playing to the alpha. And the alpha is not always the smartest in the room.
From the UK side, I think it depends on the chair very much. And if you have a good chair, they will bring people in, even if they've not been talking to it.
Right.
Because they know they've got skills in that area, and they will invite other comment, especially if there's a bit of a discrepancy between two people with a score, or as you say, an odd score. They'll bring someone in with I
would love to know if there's been a study ever done on large versus small groups in terms of looking at the same sets of grants, and do they fund the same grants? I mean, I know, for instance, I don't know if you've seen it when you're looking at graduate students. I know that if we have, let's say, 7 faculty looking at the graduate students, and then we all look at everybody, let's say we get 50, we all seem to pick the same 7. Right? Now, that's interesting to me. So do we need 50 to choose the same 7? 50 people to choose the same 7? So I would love to see if there's been any kind of statistical analysis done on something like that.
I'm just thinking about this. I'm a couple of grant panels actually currently at the moment, so going through lots of different things. I tend to be a really generous scorer and a really bad scorer. I really try and pull them apart. Yeah, the ones that are good, I will give them
I'm the same way as you.
Score.
I'm the same way as you. I'm very forgiving. I mean, I am the best person to ever get your grant or paper to review, because I generally see the merit in what people are trying to do, and I would rather spend the time drawing it out than dismissing it outright.
Yeah, but I do give bad scores too.
Me too. Oh, no. If it's bad, it's like, okay, this is hopeless.
How critical is your feedback? Mine is brutal, because I think you've got to be honest. You've got to say, if it's a low score, be nice with it, but be honest, because then they can correct it.
Yeah. I mean, unfortunately, with the NIH, you don't really give, if a paper, if a grant is not fundable, they generally don't even get they don't even get discussed. So therefore, any comments that you might have even written are never
go back. No, so UK is pretty good, and actually, even some of the American ones, I haven't seen what the feedback process is yet, but we'll see. I can't say too much at the minute.
A lot of the private ones, Peter, they won't do it either. They just won't spend the time, because they'll get hundreds of grants, and people will just sort through it and go garbage. Oh, this is, maybe this is going to be a must.
I would love to know actually, I'm also, sometimes I could be more critical of the ones I top rank than the ones that aren't, just because you think, well, it's going to get funded, but they can be so much better if they do. It's still great. Right. You suddenly realize if they read these comments, they think the score is going to be really bad. And you end up having to justify why you're saying bad comments. Did you find the same?
Right. No, it's generally, for me, it's not so much the experimental protocol that they've done, it's how they presented it. And so generally my comments are around, you need to put this here, and you need more of that there. I mean, experimental protocol, yes, if there's something, but I don't know about you, but more, I think too often, for instance, I'll be given an immunology grant to review, and I'm not an expert in every form of immunology. And so I don't have anything to tell them whether or not there should be a suppressor macrophage, or they should be using this antibody or that antibody. It's just not my domain. I'm more the big picture and how to present the idea. so that it's fundable.
Yeah, the pitch, as it were. We are nearly up to an hour. I did say it would go really fast, but I want to ask you, what is the next challenge, the next big thing that will happen that is possible? And also, if anything was to be possible, what would you like to see developed?
So I think, well, both of the things that I'm involved with are, I think, both possible and next. And one is an atomic imaging approach. And that's actually reading the position of every atom, both mass and position in an object. So that's one. And then the other is the use of basically novel physics, quantum entanglement approaches to measure things at a distance. And so I actually presented an idea, oh gosh, must have been about almost 8 years ago now, to a Nature conference of how to use quantum entanglement, or how we should be using quantum entanglement. I mean, I'm not a quantum physicist, but I thought, I think it should be possible to do this. And then lo and behold, 3 or 4 years later, somebody came out and showed that it was possible, right? To basically measure. So the idea is that you would quantum entangle a photon, send it into a cell, have it do something, and then the information you got back was similarly quantum entangled. And that way you could only receive back the information that you put in, not noise, right? Because noise is not similarly entangled. And so it doesn't read back in the detector. And so this is now being used in the military with something called quantum radar as a way to read out things at a distance. So you can see a plane at a distance because you're only receiving back the information you sent out, and they can't spoof you by sending the same kind of radio waves back because they're not entangled. An entangled photon is basically a barcoded photon, right? And so you only read back the barcodes that you sent out. So it ends up being, and there's a couple of papers already been published now on this kind of approach, and it's just the signal to noise is extraordinary. And I think with the right tools, one's going to be able to basically measure things at a distance without touching them.
So I was going to ask if you ever worry about not having another brilliant idea, but obviously that you've just answered that question within so your mind is already working out how to apply these.
The horizon, right? I mean, I've been reading science fiction since I was 8 years old, and everything from aliens to warp drive to all kinds of fancy creative imagination that people come up with. And it's you're sort of, you've already been shown the horizon.
I've got to ask 2 more questions, because you moved your head to the side for 1 minute and I saw some dumbbells, I think.
Oh yes.
What weight are you curling at the moment?
Only about 40 or 50. Nothing,
Is that pounds or kilos?
Oh, pounds.
Thank goodness. That's still a fair bit, if my maths are right, to be curling on each individual.
Yeah, 30, 40. That's the limit. It's all I need to do. It's not about bulking. It's just about, for me, it's just about, actually I've had a bad back, since I was 30 or so. And so as long as I stay fit, it's not a problem. I don't, my back doesn't go out, because when your back goes out, it's 2 weeks.
Yeah. I can empathize for sure.
Yeah.
So is that the only exercise you do with the weights?
I have a oh, well, no, I do all kinds of stuff. I have a trainer online 3 times a week. And he puts me through my paces. And then I ride my bike.
Okay. And the one other comment is, just before we started talking, you mentioned UFOs.
Mm-hmm. Yeah. So I don't know if anybody's ever seen if you've seen all the stuff on the fighter pilots around the USS Nimitz and the UAPs, right? The UFOs. It's been all over the news. I mean, if you haven't, you've been living under a rock. And the military has admitted that these things, they don't know what they are, but they're real. So I actually work with the team who got those videos released from the Pentagon. And I've been involved with this for the last 10 years since basically the CIA showed up in my office and asked me to help them with some of their pilots and ground personnel who'd gotten too close to what they claimed were UFOs, and that they'd gotten sick. And they had asked around, who's the guy who knows the most about blood analysis? And they said, oh, you need to go talk to this guy, Garry Nolan. He's got this instrument called a CyTOF. And so they showed up in my office unannounced one day, and they started showing me the data. And I honestly thought it was a candid camera joke until it started getting serious. And now it is. Just go look it up. People say, oh, Gary's drank the Kool-Aid, et cetera. No.
And
I'm still involved with it.
Is there any can you pick up immune signatures or changes with these pilots?
Unfortunately, it had happened too long ago for it to happen, but we've actually got some work ongoing where if these things happen again, we'll be able to collect the blood more immediately.
So do you think these UFOs are, natural to Earth, or do you think they are?
No idea, and I just don't know. All I'm about is collect the data. Don't ignore the data. Come to no conclusions, because if you come to a conclusion,
You'll bias it.
Wrong, and then you're debunked. So just collect the data.
Okay. I'm glad I asked that last question. That was brilliant. Garry, we're over the hour, but thank you so much for joining me today.
Yep.
Thank you everyone for watching or listening to FlowStars. So I was Peter O'Toole. Please do look at the other episodes that are out there and subscribe to the channels. Garry, thank you very much. That was brilliant. Thank you.
Thank you so much.
Bye.